No, I'm Yanlin Zhang. I'm currently a PhD candidate at McGill University, and I'm very glad to have this opportunity to introduce our recent update on the exploring 3D genome with augmented reality. In this work, we developed a mobile application. It allows users to interact and realize a 3D genome in real life. And as we are living in a three-dimensional world, the genome inside a cell also has preferred 3D structures. At different scale, it has different structure preference. For example, we have topologically associated domains. Understanding such structure preference is very important because they play essential roles in different types of biological process. For example, they facilitate the interactions between an enhancer and their promoter. Such interaction is very important in the process of transcription. They also play essential roles in the development of cells. For example, small changes in the topological structure can have a significant impact on the health of an individual. There are two there are two primary categories of approaches that we can use to study the 3D genome structures. The first one, or the direct approach, is we can use a microscope to take a snapshot of a genome, and then we can find the coordinate of several pre-select regions. This approach is very complex and expensive. It is impossible to extend this approach to capture the whole genome structure. And the other approach, or more widely used approach, is we can use a sequencer to facilitate the analysis. In this category, the most widely used approach, I believe, is a high-C experiment. In a high-C experiment, we can capture the 3D genome structures at the whole genome levels and at very high resolution. But comparing to the imaging approach, we can give the direct evaluate of the coordinate of the 3D structures. The high-C based approach can only give a contact map. In the contact map, we can know the spatial closeness of the pairwise fragment of the of a parameter. If we want to get the 3D structures, we need to use computational approaches to search for a structure that satisfies the given contact map. As the high-C is a widely used approach in 3D genome study, so there are two categories of the data representation and the realization. A more widely used approach by the research community is we want to represent the 3D genome with a contact map. If we have enough domain-specific knowledge, when we looking at the when we look at the contact map, we can imagine the 3D structures. There are different kinds of tools for the realization and for the precision or annotation of the 3D structures. For example, the precision of topological associated domain and the precision of chromatin loop. But for a person who do not have enough who do not have enough knowledge in this field, it's very hard to let us to let the person try to imagine the 3D structures by just looking at the content map. So a more direct approach to realize the 3D genome structure is we can use a 3D polymer to represent the 3D model. In order to get the polymers, we can use results from imaging experiment, or we can do the reconstructions from a high-C based approach. For both of the two representations, there are different kind of softwares we can use to do the realization and to do the analyze. In the past decade, researchers have made significant contributions in the understanding of the structures and the function rules of the 3D genome. But the concept of 3D genome is rarely introduced in a classroom. So suppose we have a biology class, and the teacher want to introduce the concept of the 3D genome. So if the teacher represents the genome with a contact map, there must be at least one student ask, where is the genome? Yes, where is the genome? We don't have a genome, we only have a heat map. The teacher can choose to use the structures reconstructed from a high-C experiment to introduce the 3D genome. I believe with this way, most students will agree with the teacher that we are working on 3D genome. But there may be at a very low probability, a student said, or a student want to challenge the teacher. It is a 2D genome instead of 3D. And this funny situation motivates us to think about the question, what is the best approach for us to represent the 3D genome if we want to introduce it to a person who do not have enough knowledge in this field? And and a very easy and mature solution that came to our mind is we can use augmented reality. This technology is indeed widely used in the intersections between science and education. For example, a medical student can use an AI application to discover the structures of our body, and the visitor can use the can use the AI application to interact can interact with a giraffe in a science museum. So here we introduce ARGV, augmented reality based 3D genome waiver. It is simple and easy to use 3D genome waiver. It allows you to run it on most mobile device. And we also prepared a large amount of dataset for users to explore and to play with. We provide more than 350 3D prompting structures, and for each of the prompting structures, we provide the topological associated domain annotations and also the AB compartment annotations. And at the species level, we also provide the annotations for prediction of genes and for the prediction of disease causing loci. So if a user use our software, and the user can interact with all of this data very easily by just click save button on their mobile device. To study the usability of our tool, we ask a class of graduate students to try our proposed software, and we ask them to conduct their 3D genome-based research by using our tool and their content map-based tool. And according to the feedback, most of the students think their VR mode actually is a very natural way to help the users to explore their 3D genome structure, and compared to the 2D based realizations, it is more user friendly, and it can help the user have a better understanding of their 3D genome organization. And indeed, more than 80% of the students think our approach helps them to better appreciate the importance of the 3D genome structure. And I believe when introducing a new software, despite how many slides I show and I talk, it is not interesting. And the best approach is the best way to introduce a software is to give a very short demo. And our principal contributor to the development of software, Chris, will lead a tour.
Welcome to ARGB, an augmented reality genome viewer. The following videos will demonstrate the features of this application. Part 1, Overview. This section includes an overview of the main menu, database selection, and augmented reality mode. Main menu overview and selecting the genome viewer mode. The main menu consists of selecting either the mobile regular mode or the augmented reality mode. You can also select any database functionality from the top right of this section. Database Downloader, downloading datasets from the server. From the top right of the main menu, select Download. This loads the database downloader section. First, select the collection you want to explore. This will load all the datasets you can download from that collection. Select the dataset you want and click on Download. In order to explore it, with a large database of over 350 datasets to choose from in one convenient location, Augmented Reality Mode. Scanning local environment to set up Augmented Reality Mode. To start AR mode, select Augmented Reality from main menu. Scan local environment by moving mobile device around. When scan complete, tap screen to place marker where the 3D model will be placed. The now be enabled in augmented reality mode and can navigate with it in real 3D space. Part 2, Genome Selection Settings. How to enable selected model and any annotations. Genome settings, basic settings, selecting chromosome type. Updating model can be done by selecting the species, assembly, cell type, model set, and chromosome. Then click on Set Chromosome to enable your selection. Genome settings, advanced settings, enabling genes and annotations. Advanced chromosome settings can be found on the bottom right of the screen. Enabling genes, activate gene visuals. To toggle genes on, you must select the Gene tab on the bottom right section of the screen. Then wait for them to finish rendering to explore them in detail. Enable annotations, activate annotation visuals. From the bottom right of the screen, click on Annotations. Then select the annotation you want to enable by toggling it on. Wait for them to finish loading, to then explore them in more detail. Annotations in the compartments, special annotation type. Similarly to regular annotations, open up Annotation section, then select specific annotations labeled with an underscore, a OB at the end of its name to load this specific type of annotation. You can now see a special type of highlighting for these specific type of annotations with section off transparencies. Annotations tabs, special type of annotations. Select Annotations, then find an annotations that ends with tabs and click Enable. Wait for annotations to load. This annotation visuals are different, having alternating transparency sections to distinguish each segment. Data panel, detailed info for each selected region of interest. When selecting a region of interest, either by clicking on region of chromosome or searching, a data panel pops up with detailed info for this region. You can then see a table and graph associated for this model for all annotations and genes in the selected segment. You can switch between annotations on the top right of the panel and expand each section as needed. Part 3, additional features, search, share, bookmarks, and more.